busy day in the clinic, guys. How's your lunch going?
Yeah, it's really good.
That's good. I'm sorry to interrupt, but I have an interesting question regarding one of my clients. I just want to get your advice on it. I have a client from rural northern Australia who has a canine stud and is wanting to collect semen themselves and sell it overseas. They don't have access to a vet close by, so they're thinking of collecting the semen themselves and then transporting the sample to a facility that does the freezing. Do you guys remember that study that we looked at from that conference a few weeks ago that was about this?
I do, yeah, I remember that one. It was asking this question pretty much exactly, the paper by Colombo et al. Yeah, it assessed the freezability of ejaculated canine spermatozoa after 24 and 48 hours of cooled storage in field conditions. And then did they do a secondary experiment to assess the effects of different extenders?
Yeah, it was that paper from the conference 2 weeks ago. Well, it's really important to understand the implications of delaying sperm freezing times to allow for transportation of semen samples. The freezing of sperm requires special laboratories. However, the breeding studs, like in your case, Indy, can be located far away from these facilities, making it difficult to transport samples without compromising quality. Investigating ways to successfully cryopreserve semen in the field is essential for many reasons.
Yeah, and a better understanding of this may not only allow for the transport of cooled semen in domestic animals such as a canine, but the research could also be expanded to endangered species. This study can help avoid issues such as inbreeding, as it allows different genetics to be transported farther across distances. So a wider range of genetics is accessible to different studs and breeding facilities, allowing for an increased genetic diversity.
Yeah, and the ability to freeze sperm can optimize inseminating doses and tighten up the window of genetics, as one stud sperm can be inserted into multiple females. An example of this would be the selection of better genes that would provide ideal traits such as good recall, higher intelligence, and more obedient working dogs for fields such as the military and the service. Also, through these methods, direct contact between animals can be avoided, which decreases the risk of sexually transmitted diseases.
Yeah, an important consideration when undertaking a canine semen cryopreservation study is having the understanding of species-specific anatomy and physiology. As we know, assessment parameters can include volume, color consistency, motility, both total and progressive, morphology, concentration, and acrosome integrity, etc.
The ideal canine semen consistency is dense and thick, whitish in colour, and the ejaculate contains a concentration of about 100 to 300 million sperm per mil.
Yeah, and seen in this picture, the normal morphology consists of an intact plasma membrane, a head with the nucleus and acrosome, the neck, the midpiece with the mitochondrion, and the tail. In the study that we looked at, the structure abnormalities in these structures included things like a coiled tail and a bent neck or midpiece.
It's also important to notice that canine spermatozoa plasma membrane has a high cholesterol to phospholipid ratio, which means they're semi-resistant to cold temperatures, so cryopreservation is a possible technique for semen storage. In contrast, raw semen is much more labile and therefore cold sensitive due to higher protein and lower cholesterol content. With this in mind, canine semen can act as an excellent model to explore the effects of standing time prior to freezing.
Okay. So in the study we saw at the conference, 20 healthy and pubertal stud dogs were used, aged between 2 and 13 years. They range between 4.6 to 60 kilos in body weight, and the breeds used were unspecified. However, it's noted that there were varied breeds used.
Yeah, and in experiment 1, they collected 10 semen samples and divided them into 4 aliquot samples to compare semen viability when frozen at 0, 24, and 48 hours. Experiment 2 was similar in this sense that they had 4 different aliquots, 2 with a chilling and a freezing extender at 24 and 48 hours.
Yeah, the study also used an array of relevant statistical approaches to determine the significance and reliability of the results. The mean, while the mean value is important to gauge an average of the results, it doesn't take into account the high diversity of subjects used in the study, so standard deviation was also used. Standard deviation is good because it shows how much the members of the group skew from the mean, showing any outliers, which in the study is good because a wide variation of breeds, ages, etc. were used. Kolmogorov Smirnov test was also used, and this compares data to a known distribution, in this case the World Health Organization, to indicate that there's a similar distribution in results. Krushville test used to compare the statistical significance between the 2 experiments where there are 3 or more groups, i.e. the multiple freezing types and extenders used. In the study, the p-value was also set at less than 0.05 to determine an accurate statistical conclusion. This is an unbiased way for researchers to reject or validate the null hypothesis. So overall, I think these statistical approaches were ideal with the methods used.
Yeah, so experiment 1 found that freezing at 24 and 48 hours from semen collection did not significantly affect the post-thaw quality. As expected, morphology quality of fresh control was better than that of frozen samples. The experiment was compared to the World Health Organization's data, which indicated that morphological indices increased after freezing regardless of freezing time, especially the sperm deformity index due to the abundance of acrosomal defects found during morphological evaluation. This trend was confirmed in the study where intact aprosomes were higher in fresh samples and lower in frozen ones.
Results of motility show that pre-freezing motility was lower after 48 hours in the transport box, but there was no statistical difference found in post-thaw motility among frozen groups. The ability of spermatozoa to bind to the zona pellucida of frozen oocytes was not influenced by freezing, nor by pre-freezing holding time in the transport box.
Yeah, and looking at this table, it can be seen that motility pre-freezing was overall significantly higher when thawed versus post-thaw, so like 86 to 11.3, like that's quite a significant difference. And no matter what, motility at 48 hours was significantly less, with a mean of 58% than that of 0 and 24 hours.
Yeah, I mean, wow, these results support the literature. Cryopreserved sperm are shown to undergo precocious capacitation. Capacitation results in hyperactivated motility, which causes the sperm to expand their energy reserves, reducing their progressive motility and overall survival rate, therefore lowering fertilizing ability.
Experiment 2 results showed that morphology was not affected by the extended type or freezing time in this experiment. There were no differences found in the different indexes. Motility at thawing significantly decreased at 48 hours compared to 24 hours when the chilling extender was used for dilution, but remained comparable in most of the results obtained at 48 hours when using the freezer extender as a diluent.
So you can see here that in experiment 2, where the freezing or chilling extender was used, there was no significant difference in motility from pre-freezing to motility at 6 hours post-thawing. However, compared to using no extenders in the experiment 1, the motility, even when sperm were frozen, immediately decreased from, as you said, 86 to 11.3 from pre-freezing to 6 hours post-thawing, which is quite significant.
Yeah, so overall, it can be concluded that these results were successful in meeting the aim of this assignment.
That's great. Well, results drawn from the study are clinically significant, especially looking at motility and ultimately the ability for sperm to travel through the female's reproductive tract. Spermatozoa must travel, depending on the method of AI, through either transcervical or intravaginal, through the cranial vagina, the cervix, the uterus, and then the uterine horn to fertilize an egg.
Yeah, and an interesting point to make too, if sperm motility is dramatically decreased, like showed in this experiment, could intrauterine insemination be an option to decrease the distance needed for the sperm to travel to fertilize an egg? Because the cervix is one of the main barriers for sperm to travel through, and it contains goblet cells which can secrete mucus in the cervical crypts to flush out foreign materials such as the sperm itself. The experiment has numerous flaws that, if improved, could significantly affect these results.
Yes, there were a number of flaws. A very small subject size of 20 dogs was used, increasing the margin of error in results. To add to this, the subjects used ranged greatly in age, breed, and weight, with the oldest dog being 13 years old. We know that semen quality decreases significantly with age. A larger sample size would greatly improve the accuracy of the study. However, being the first of its kind, it creates a pathway for future studies.
Yeah, I agree. Also, it wasn't mentioned if a control period, if there was a control period before sperm collection occurred. So my question is, were the dogs monitored for a minimum time of the epididymal transit time, which is 12 to 14 days of the dog, and as well as the spermatogenic cycle, which is 62 days, to ensure that they did not endure fever or physical trauma to the testes and things like that. The testes must be made at a temperature 4 to 6 degrees below body temperature, so any event like a fever can impair sperm viability.
Yeah, the dogs were also noted as healthy. However, the author has given no parameters on this definition of this term. What would indicate a healthy individual is very subjective, and we know sperm production and quality has a positive/direct correlation with the health of an individual. For example, factors such as obesity or parasite burden, etc., can negatively affect the sperm quality and can skew the obtained results.
Yeah, definitely. Also, in this experiment, it was stated that the transport box was kept at a winter ambient temperature, but an exact temperature wasn't provided. So, if the same procedure was conducted in the summertime, the environment of the transport box may be significantly warmer, which basically counteracts the point of the cooling transport box.
Is it not important to factor in that the ice packs will melt and how can they keep these frozen for the holding period? This wasn't mentioned.
Yeah.
No, and that's a very good point, and these are all good points to keep in mind. Also, in both experiments, it is not specified whether the assessment was done by one consistent person or multiple people, where the results may have been interpreted differently.
Yeah, and each experiment only used 10 semen samples, which again increases the margin for error in the results. Ideally, semen sample size should be increased to gain a more accurate representation of morphology and motility.
And lastly, it is important to mention that the sterility of the experiment conditions was not noted. Dirt, dust, pathogens, etc. will impact sperm motility and quality, and if not carefully avoided may impact the sample and therefore the results of the study.
Yeah, and the source of these subjects used for the study was not confirmed. Was it the same breeder or were they acquired from different studs? This could play an important role in the results obtained, as the husbandry and management of one stud may vary greatly to another stud.
Yeah, and given all these flaws, it can be assumed that because the study was funded by the Polish National Agency for Academic Exchange, which is a company that stimulates research for academic exchange, the agenda and the results for this study were not distorted to favour the funder. Well, it's nice to see that overall the outcomes of this study are widely supported by research and other studies in this area. Firstly, when you look into motility, which was the most significant result, 3 separate studies by Domain et al. And Payne and Forsenberg and Silver et al. All found that motility was improved in the fresh samples as opposed to those chilled and frozen, and concluded that any post-thawing of sperm affected motility when compared to fresh semen.
The present study also found post-thaw sperm quality dramatically decreases. Chatterong et al. Supports this as they found that all of their parameters that they measured, like motility, acrosome, and DNA integrity and viability, dramatically decline after 6 hours.
Many previous studies like this one also use extenders to assess if this has had an impact. Hewitt et al., as well as the above mentioned, also found that freezing sperm with extenders maintained motility. A study by Dira et al. Was super interesting in that they found that the variations in the sperm head area depended on the cryopreservation procedure. They found conventional freezing decreased the head dimensions, and vitrification led to an overall increase in the sperm head size, which ultimately affects fertilization.
Yeah, wow. This research is interesting and it could be incorporated into further studies of in-field cryopreservation to see if it's possible to freeze semen using the vitrification method without laboratory equipment.
There was some new knowledge gained from the study. Previous studies have focused on the freezability of dogs' spermatozoa, but the present study is the first to undergo dilution and preservation in the field. They found that a major parameter of statistical significance during in-field cryopreservation was motility. So the next step really is to figure out a way to improve spermatozoa motility at the wing.
Yeah, and due to the high content of polyunsaturated fatty acids, canine spermatozoa are sensitive to reactive oxygen species. This is something that must be kept in mind when creating an in-field canine semen cryopreservation technique, because the buildup of reactive oxygen species leads to oxidative stress and attacks polyunsaturated fatty acids in canine sperm plasma membrane. Reactive oxygen species are created by spermatozoa themselves during storage in an aerobic environment, and it has been found that their levels increase after cryopreservation.
So you're saying that if the sperm is subjected to a longer stay of time in the field, reactive oxygen species may accumulate, decreasing sample quality?
Yeah, and to combat this issue, some researchers have even been looking at cryopreservation techniques that incorporate antioxidants to limit reactive oxygen species levels. I found a study which found that the addition of antioxidants improves the quality of dog semen, especially with the addition of vitamin E, B16, taurine, which all show to increase or improve progressive fertility.
That's really something which is going to be super interesting for future studies. We know theoretically that in-field cryopreservation is possible, but this technique will need to be tested in AI experiments before any final conclusions can be made. Other studies could also potentially look at using different transport mechanisms. The freezer box used in the study isn't the most reliable, and it's difficult to keep a constant temperature, but some studies were successful with ice fridges, so maybe a portable car pilot fridge could be a possibility.
Well, overall, for these types of studies, it's interesting that small sample sizes seem to be commonly used. Are they struggling to find owners that are willing to do this, do you guys reckon?
Yeah, and is the reason for only using stud dogs because of like good genetics?
But why would you, for study purposes, not look at collecting samples from the RSPCA to get a greater sample range? Why would you limit your study to only using genetically fit sperm when you could use a wide variety of samples and subjects?
That's such a good point, and I guess the reason could be because of the time cryopreservation is used for breeding animals who generally have desired genetics. But I do see what you're saying. If we were to use a wider variety of animals to get a true accuracy of semen preservation, that may provide us with a better understanding of the pros and cons of semen cryopreservation.
Yeah, and then once we have these results and they've been identified, they can be implemented on these stud animals and put into action.
Yeah, I completely agree. It's also interesting to note that studies on cryopreservation and related topics have been studied for nearly 20 plus years. Numerous studies have looked into this area, and it's interesting to think about why there's been very little advances in the past 20 years.
Yeah, and the common trend between these studies showed that overall motility and morphology are negatively affected during cryopreservation, especially semen being thawed. But why has there been no advances in sperm storage techniques?
Yeah, like maybe there's a cost at play. Like when you look at the ultra freezer used in Almer et al. Study, it was probably incredibly expensive. Not enough demand and research into the storage aspect maybe. But with regards to vet practices like ours, further research into this topic would potentially lower the cost of owners in sending off their samples. I think you could potentially gain a greater client range in offering this through vet practices. This could also help with increasing the gene pool, not only using stud animals who live near the semen freezing facilities.
Yeah. And I think lastly, it's important to touch on we're in the current climatic of zoonotic diseases such as COVID-19. These results show that there may be a valid solution to performing collection at the start by breeders themselves and then shipping or posting sample off to a nearby facility for freezing. This may prove useful in times of limited social contact and or isolation to reduce the movement of animals and people and therefore the spread of disease.
Yeah.
Oh, excellent guys. I definitely have some great points to bring back to my client. Thank you so much for this discussion.
Thank you.
Thank you.